Hi, class. So this video is going to cover DNA, RNA, protein synthesis, and meiosis for Bio 121. These topics are covered in parts of chapters 2 and 3 in your textbook. I've listed the sections and the page numbers in D2L. So why are we having this? Why do I have to make another video? Why do you have to hear another video? Because in our syllabus, we have these things called measurable performance objectives, or MPOs. They list all the things that must be covered in the class. So the ones we have not yet covered are to describe the structure of DNA and its significance, and the significance of complementary base pairing, describe RNA structure and name the 3 types, describe transcription, translation, and the function of mRNA and tRNA, and describe meiosis and its significance. So that's what this short video is going to do. We're going to do this very light and just to give you enough information so that you can better understand fertilization and the hereditary aspects of chapter 29. Oh, and also define haploid, diploid, and relate them to normal chromosome number. All right, so starting with DNA and RNA, they are both types of nucleic acids. DNA forms the genetic code in the nuclei of body cells, and it regulates most of our cells' activities. RNA guides protein formation. So RNA is really strictly just with proteins. All nucleic acids are made of chains of nucleotides. One nucleotide is made up of a pentose or 5-carbon sugar, which can be either ribose or deoxyribose, a phosphate group, and a nitrogenous base. The nitrogenous base can be adenine, guanine, cytosine, thymine, or uracil. Here we see what a nucleotide looks like. Over here, we have a pentose sugar. They can be deoxyribose for DNA and ribose for RNA. So just as a reminder, DNA is deoxyribonucleic acid. So it's a nucleic acid. RNA is ribonucleic acid. Okay, so here we have a pentose sugar. We have a phosphate group stuck on there. And nitrogenous bases can be any of these 5. For DNA, we can have cytosine, thymine, adenine, or guanine. Okay, and we abbreviate them CTAG. For RNA, we do not have thymine. Instead, we have replaced it with uracil. So they can have the 4: cytosine, uracil, adenine, and guanine. Now in the MPO, it said something about understanding complementary base pairs. Well, a strand of nucleotides, which is a nucleic acid, can line up with a complementary strand because the little parts that come off of that backbone have the nitrogenous bases. So imagine this side being a line of a bunch of these just stuck on each other. The nitrogenous base kind of sticks out towards the middle. If this nitrogenous base was, say it was cytosine, on the other side, there could be a hydrogen bond here, and it would bond cytosine to guanine. So C and G always are the complementary base pairs. Adenine will bond with thymine in DNA, or in RNA, we don't have thymine, so we have uracil. Okay, so A and T or A and U. Let's look at DNA. So DNA is 2 strands. It's considered a double-stranded helix because it loops around like this. Here is that backbone I was discussing, and off of it are those nitrogenous bases. You can see T, hydrogen bonds to A, so thymine and adenine. C, hydrogen bonds to G, cytosine and guanine, okay? And here we see a close-up of that. So if we break it down in the middle, the left side here is our backbone, which is the purple part. And then coming off of it are these nitrogenous bases. Okay, and the same on the opposite side. We have that purple backbone, which is going to be these 4 things. And then off of it is the nitrogenous bases. And you see T bonds with A, C bonds with G. So some more things about DNA. Each strand is a bunch of nucleotides bound together. Oh, what else? We went through the bonding, and that's about it. That's all that says. Now, this is a good chart showing the differences between DNA and RNA. So the bases, A, C, and G are all the same. Thymine is in DNA and uracil in RNA. The sugar, the 5-carbon pentose sugar, deoxyribose for DNA, ribose for RNA. DNA is double-stranded. RNA is only single-stranded. The base pairings, the same except for the T and the U are switched. How is it copied? DNA is self-replicating. RNA uses DNA to make a blueprint. The functions are different. So DNA encodes the information for making proteins, and RNA carries out that information to make the actual proteins. There's 2 types of DNA, nuclear and mitochondrial. RNA, there's actually 3. Your book is just discussing messenger and transfer, those 2, but there's another one called ribosomal rRNA. We're not even going to go into what it is, but just know there's 3. mRNA is messenger RNA, transfer RNA is tRNA, and ribosomal RNA is rRNA. So how does RNA and DNA make proteins? So we already learned in Bio 100 that, or 120, that most of our most of our body, we have all these proteins. We have tons and tons of proteins. So how are they made? Protein synthesis. Protein synthesis is made up of 2 parts. The first step is transcription. The second step is translation. Transcription, what ends up happening is this is supposed to be a cell. This is the nucleus. Inside the nucleus, as you should know, is our DNA. So inside of this cell, we have all this DNA, and it codes for what the proteins are supposed to have. But to make proteins, you have to have a ribosome, and they're not located inside of the nucleus. They're located in the cytoplasm. So the DNA information has to get out here to a ribosome. But DNA cannot fit through these small pores in the nucleus. So, because it's double-stranded, it's bigger. So instead, what it does is it creates a copy of this information into the mRNA, okay? So transcription, all it's doing is taking the DNA and creating an mRNA, okay, messenger RNA. That messenger RNA is only single-stranded, and it can leave the nucleus through one of these pores and go out to the cytoplasm and find a ribosome and then make a bunch of proteins, okay? So transcription is inside of the nucleus. Translation is outside the nucleus in the cytoplasm where there's a ribosome. All right, so transcription, part 1, occurs in the nucleus. It's the process where the genetic information coded in DNA is copied onto mRNA. Okay, so this shows the whole process. You don't need to know that. This just shows inside the nucleus, we have this enzyme. The purple thing is the double-stranded RNA or DNA. And then the red is the mRNA that is being coded for, that is created. Okay, so they make this mRNA. And that is transcription. So, I'm getting rid of this slide, so you don't need to know this one. Translation is the second part of protein synthesis. It happens in the cytoplasm on a ribosome. The ribosome reads the mRNA, and then it determines the amino acid sequence. So we have to have amino acids, amino acid, amino acid in a line bonded together, and that is a protein. So what is the sequence of them? Well, the ribosome reads the mRNA to determine that. As it reads it, and it's like, oh, the next amino acid is such and such, transfer RNA brings the correct amino acid over. Okay, so that's the job of transfer RNA. It brings the correct amino acid to the ribosome as the mRNA is being read. The amino acids are then bound together with peptide bonds, and that forms the protein. Okay, here's the whole process of transcription, and note you will not be tested on these steps. Okay, so this just shows This peach and purple part, this is the ribosome. It has two parts to it. This strand right here, that's the mRNA. And here's a tRNA bringing the correct amino acid. So the ribosome reads the different nucleotide base pairs. And then, oh, we need this amino acid. So tRNA goes and gets it, and it comes and brings it in. And then all of those, here we go. All of the amino acids, they end up being bonded together with peptide bonds. And eventually you end up with a whole strand of them like this. All right. Okay, now getting to mitosis and meiosis, is the last part you guys need to learn. So just as a reminder, we went over mitosis in AMP1, and it's just the process of making exact replicas of a certain cell. So say you got hurt, you got a cut, and you need to fill in that gap with more arm cells or finger cells, skin cells, whatever. Your body's gonna use mitosis. You're going to have one cell, it's going to go through mitosis and give you two of the exact same cell. So the stages of mitosis, if you remember, was PMAT, prophase, metaphase, anaphase, telophase. Okay. Now, a cell that has 46 chromosomes, like most of our body cells, is called 2N, or it's known as diploid. Okay. So in mitosis, we just make more cells with 46 chromosomes, more diploid cells. Okay, so this was a diploid, these both are diploids because they're exactly the same. So every cell in our body is diploid, except for gametes. Gametes, if you remember, are sex cells. So ova or secondary oocytes for females and sperm cells for males. They cannot have 46 chromosomes because when they fertilize, it would be a 46 and a 46. They come together, that would be 92. And then we'd have this weird offspring, okay? So in order to continue on the genetic race of humans, we have to have, and all animals go through this, we have a haploid number for our gametes, okay? So we have to make some oocytes and some sperm with only 23 chromosomes in them. And to do that, we use meiosis, okay? This is reproductive cell division. This is how we make haploid gametes. It's a two-step process, and in it, we have the number of chromosomes from 46, which is everywhere else in our body, which is a diploid number, to 23 chromosomes, which is a haploid number. This is important, so when fertilization of the secondary oocyte by a sperm occurs, it will result in an embryo with just 46 chromosomes and not 92. So if we had two of these, 246 coming together, then we wouldn't end up with a viable offspring, honestly. So in meiosis, one diploid cell ends up making 4 haploid cells. So I said there was 2 parts to this. The first is meiosis 1. This shows it on the left here. Before we even start this, the cell has already made a copy of its DNA during the interphase process, actually the S phase of interphase, if you remember that. So the first part of meiosis 1 is prophase 1. In this phase, the chromosomes condense, but they also pair up. The chromosomes align with its partner, so the genes on each chromosome are right next to each other. They're lined up. Then a very important thing happens. There is what's called crossing over. While these 2 chromosomes are lined up, sections of the pair are going to exchange places. So when the chromosomes are lined up, the genes on one chromosome for a specific quality is going to be lined up with the same gene for that quality on the other chromosome, and they can switch places. This accounts for all the genetic diversity we have. That's why my kids are not exactly the same as me. Okay, so my secondary oocytes, they're going to have combinations in all of all of the different oocytes of these crossing overs because of these crossing overs, okay? After prophase 1 occurs, metaphase 1s occur, and here the pairs of chromosomes line up. Anaphase 2 is the homologous chromosomes separate, but the sister chromatids are still attached. In telophase 1, the chromosomes decondense, cytokinesis occurs, and you end up with 2 haploid cells. Now you're going to say, how is this haploid? I see 4, like I see more than I had. It's considered haploid because there is only, the diversity is different, okay? We still count them the same, but let me show you in meiosis 2, they're going to be the same number of sister chromatids as there are chromosomes here, okay? So best case, just know that 2 haploid cells are formed here. I'm not going to get into asking questions about the specifics of that. So meiosis 2, you can think of meiosis 2 as mitosis for haploid cells. So we're starting off with 2 haploid cells, you'll see these, and we're going to end up with 4 haploid cells, okay? Because each one creates 2. So in prophase 2, the chromosomes condense, the spindle forms. Metaphase 2, the chromosomes line up. Anaphase 2, the sister chromosomes separate. And telophase 2, the chromosomes decondense, cytokinesis occurs, and 4 haploid cells are formed. Okay, I apologize, these should all be 2s. And that's it. So now you can go on and finish your last chapter, which is chapter 29. Thanks.